Hello, my name is Brigitte Baella, and I am a senior from Puerto Rico studying biological basis of behavior with a minor in chemistry. My Ben talk is on the epigenetic regulation and corresponding expression in the Harpagnathus saltator and Drosophila melanogaster. For the past year, I have been doing research on epigenetics with the model animal Harpagnathus saltator. I am currently in my second semester of independent research for my major. This talk will be divided in 5 parts, beginning with my involvement in this research lab. Then, I will briefly describe the basis of epigenetics as it relates to my research. Afterwards, I will explain why this animal model is used for epigenetic studies. I will go on to explain my lab's findings in my own research. Finally, I will explain why focusing on epigenetics is so important. I am part of the Bonacio Lab, which is located in the Smilo for Translational Research building. It is part of the Cell and Developmental Biology Department and the Penn Epigenetics Institute. My research was spearheaded by my PI, Roberto Bonacio, and the postdoctoral fellow, Janko Gosposic. I began my journey in this lab doing husbandry work in the ant room, mainly working with the species Harpagnathus. This included feeding, maintaining healthy colonies, marking individual ants, and studying behavior. Then, during the summer of 2018, I transitioned to performing wet lab work. During the fall of 2018, I studied epigenetic regulation in Harpagnathus saltator, as a continuation of Gosposic's 2017 study on chorozoning levels in Harpagnathus. During spring of 2019, I am currently working on behavioral analysis of Drosophila with genetically manipulated levels of chorozonin, as a follow-up of the same study. In addition to these research endeavors, I was lucky enough to receive a cover in Cell Reports for my photography work on the ants, as well as in other neuroscience and epigenetic publications. So, what is epigenetics? In simple terms, epigenetics is a change in phenotype without change in genotype. This variation can be inherited, and the molecular mechanisms that underlie this epigenetic memory are important in many processes, such as embryonic development, cancer, stem cell pluripotency, and brain function. This epigenetic information can be encoded in molecular changes of chromatin. Chromatin is a complex of proteins, DNA, and RNA that packages the genome within the nucleus. Some of these changes include DNA methylation and histone modifications. DNA methylation is a covalent modification that alters the rate of transcription. Histone modifications are covalent post-translational modifications that impact gene expression by altering their structure. Harpagnathus saltator, seen here, is a species of eusocial ants that has been a subject of investigation because of its particular behavioral plasticity. Its colonies are divided into castes according to specialized behavioral tasks. In these colonies, there exists only one true queen. She is the only reproductively active individual within a colony, and has distinct differences in behavior, physiology, morphology, and lifespan. The queen will not eat molest, she will stay close to her brood and assume a dominant position against the workers. Her main occupation is egg laying in order to increase the size of the colony. The rest of the colony is comprised of workers. Workers are responsible for foraging, provisioning food, and defending the colony. In summary, the queen is in charge of the reproductive aspects of the colony, while the workers are in charge of hunting. When a queen dies, or is no longer capable of maintaining the colony, worker ants engage in a tournament-style duel in order to replace her. These ants employ tactics such as biting and policing until various ants win each tournament. The winners, called gamergates, will undergo both physiological and behavioral changes. The gamergates will activate its once dormant ovaries, effectively becoming reproductive and laying eggs. They will cease to perform its worker behavioral tasks and employ behavioral changes that align with those of the former queen, such as an aversion to leave the nest, dominant stance against workers, and laying eggs. The transition between a worker and a gamergate occurs in adult ants and is accompanied by a change in phenotype. Given they arise from the same genotype, the emergence of these distinct phenotypes is called polyphenism. In Harpagnathus, polyphenism distinguishes castes according to their reproductive abilities. queens and gametes, which are able to reproduce, and workers, which are not. Molecular pathways responsible for this diversion are ultimately directed by transcriptional regulation of gene expression, which controls activation of certain genes or parts of the genome. A cell's ability to establish, maintain, and reverse these transcriptional patterns is due to epigenetic regulation. In order to study these changes, researchers need to be able to manipulate their genes. These can be done by creating transgenic constructs and thus altering the transcription of certain genes and neuropeptides. One such neuropeptide is curazonin. As discovered by Gosposic, curazonin is downregulated as workers become gamergates. In addition, he discovered that injection of curazonin in transitioning ants stimulates worker-like hunting behaviors, while at the same time inhibiting gamergate behaviors. In the same publication, Gosposic investigated the effects of curazonin on Drosophila melanogaster, which is a model organism for genetic manipulation. He found increased egg-laying rates when curazonin was knocked down, while overexpressing curazonin resulted in lower egg-laying rates. These findings further support the existence of a common pathway connecting curazonin with reproductive behaviors in insects, as seen in both Harpagnathus and Drosophila. Because of this, my research focuses on investigating if curazonin also regulates dominance behavior in Drosophila. These organisms, in addition to having virgin to mated transitions, have multiple tools for genetic and behavioral investigations. Some of these tools include RNA interference, sulfate tracing, and overexpression systems, which are used in order to determine gene expression. In the fruit fly Drosophila, it has been shown that virgin and mated flies display different levels of dominance behavior, with mated flies showing increased aggression. This change in behavior can be compared to the change in behavior between workers and gamergate Harpagnathus ants. Given the highly conserved pathway between curazonin and reproductive behavior in Harpagnathus and Drosophila, I propose curazonin could also be involved in changes in dominance behavior between virgin and mated flies. The goal for my independent research is to assess whether the circuitry responsible for the dominance behavior change between worker and gamergate in Harpagnathus is conserved between virgin and mated Drosophila in terms of dominance behavior. In order to explore this, two tactics were designed in order to quantify curazonin level dominance behavior. For the first goal, virgin and mated flies of different species of Drosophila that naturally show different levels of post-mated aggression will be scored on behaviors such as headbutting, leg fencing, shoving, retreat, and feeding. For example, a pair of flies, either both virgin, both mated, or one of each, are placed on vials and recorded. I score every time the flies engage in each of the dominance behaviors previously described. After all videos are scored, multiple statistical analysis will be done in order to determine if there are any significant changes between virgin and mated flies. In addition, curazonin level expression will be checked using qPCR in order to verify if the switch in behavior is due to change in curazonin expression. qPCR is real-time polymerase reaction, in which the amplification of the targeted DNA is monitored in real time. PCR is a tool used in molecular biology in order to make many copies of a specific DNA segment. In this experiment, it is being used in order to study the pattern of gene expression in order to see which genes have become active or which have been switched off. For the second goal, an inducible neuron-specific driver will be used to either overexpress or knock down curazonin in the brain of both mated and virgin flies. This will be used to see how the presence or absence of curazonin influences dominance behavior in virgin and mated flies. The tool used in this part of the experiment is the GAL4 UAS system. This is a biochemical method used to study gene expression and function in organisms such as the fruit fly. Fruit flies have been modified in order to express the GAL4 gene in certain cells or tissues, such as the brain. GAL4 doesn't affect any change until it binds to a UAS region, which is also inserted into the cells via genetic modifications. The UAS region is placed next to the gene of interest, and once GAL4 binds to it, the gene is transcribed and starts producing protein. By this method, curazonin can be either overexpressed or knocked down in specific regions, and then dominance behavior can be analyzed. In conclusion, My research focuses on the effect of the neuropeptide cortisone in regulating castor termination in the ant species Harpagnathus saltator and dominance behavior in the fly species Drosophila melanogaster. By the end of my senior spring semester, I will have an answer to the proposed question of how cortisone affects both species and how the circuitries of these two animal models are related. Why is this important? The study of epigenetic changes is of utmost importance because of its implications in a variety of human diseases. While epigenetic changes are required for normal development and health, they can also be responsible for many diseases. Since epigenetic changes, such as DNA methylation and histone modifications, can silence or abnormally activate any number or type of gene, many diseases can be triggered. Some diseases include cancer, autoimmune disorders, and neurological disorders such as Alzheimer's, Parkinson's, and schizophrenia, as well as affecting brain and memory function. By studying and understanding animal models, such as that of Harpagnathus saltator and Drosophila melanogaster, we are one step closer to understanding the epigenetic modifications in humans. The association of epigenetic changes with specific diseases can help further the development of tools used to diagnose, treat, and cure patients. As a pre-med student, I can only begin to imagine the applications of uses for this type of research in the medical field. Thank you for listening. I want to give special thanks to my aunts, my flies, and my PI, Roberto Bonacio, for helping me along each step of the way.